Okay, thanks very much for joining in on this talk. My name is Kartik and I'm a postdoctoral fellow based at the Queensland Institute of Medical Research in Brisbane, Australia. And today I will talk through a series of studies that explore the relationship between brain structure and function in children with acquired brain injuries. So I don't have any disclosures, and before we get into it, I think it's important to preface this talk by saying that children's brains are different to adults. It's a very obvious point, but depending on whether you're examining neuroimaging from younger children or in preschool or in primary school or later at adolescence, there are some important developmental considerations to keep in mind. For those of you in the pediatric imaging field, you can appreciate the challenges of imaging a variety of children, and in particular those who have attentional, cognitive difficulties, and also the balance of acquisition from a technical point of view. In general, an acquired brain injury or an ABI can interfere with a host of critical developmental processes, including factors such as neurogenesis, neuronal migration, synaptogenesis, and myelination. And we can often attribute ABIs in childhood with the reorganization or reconfiguration of brain networks. And in children, we like to view the lens of an ABI with a facet of neuroplasticity. Children can often have better recovery trajectories purely because their developmental trajectory is strong, or rehabilitation has been provided at the right time. And conversely, these trajectories can be heavily impacted if the recovery from an injury is slow or the treatment being provided is not having the desired effect. So what we often see with a range of ABIs is that an injury disrupts at either a subtle or coarse level. These disruptions to grey and white matter integrity can impair the efficient transmission of signals across the cortex and disrupt the synchronization of brain networks. And this is because following an ABI, a child's brain may attempt to compensate for the damaged regions by recruiting alternative pathways or utilizing different brain regions to perform specific functions. Although the scale of structural recovery can be slower, functional networks do tend to rapidly change. And this adaptive reorganization can help mitigate the functional deficits caused by the injury. However, it can also lead to an inefficient or less optimal network configuration, which may result in compromised cognitive, motor, or sensory performance, and that can persist for some time as well. So for this talk, we'll look at a focused case of injury, being mild traumatic brain injuries, which leads to concussion. And in children, mild TBIs occur in those who have sporting injuries or falls of a minor nature. And typically, when they present to the emergency department, assessments may necessitate a CT or MRI scan alongside concussion tests. Now, depending on the child or the type of injury or the location of the injury itself, various functional abilities such as cognitive attention, motor, and sensory functions can be affected in the days and the weeks post-injury. And what we've observed is that these symptoms can last quite a long time. In fact, they can last up to 2 years, which is quite a significant impact on a young child, in particular for their schooling or their social participation. And the common complaints of this injury, aside from attention, include things like sleep and fatigue, emotional regulation, and also declines in overall academic performance. So in this talk, I'll give you a descriptor or some of the insights that we've gained from brain imaging in this cohort, and what kind of structure and functional changes that we've observed, and some of the methodological considerations along the way. The cohort that we have focused on is MTBIs in about 100 kids aged between 8 to 18 years. Again, I'm giving you a bit of a snapshot on this cohort, so I'll go quite quickly. And they have a variety of post-concussion symptoms at the 1 month mark. They had structural T1, diffusion-weighted imaging, alongside resting state fMRI and a task-based fMRI as well. And at this 1 month time point, they also had neurocognitive and behavioural testing alongside a range of other symptom and functional profiles. And of these children, about 70 of them didn't have a full recovery and remained symptomatic, and about 30 of them did make a full recovery. And what's interesting about this cohort is that it's very typical, in fact, of what may be collected in many major tertiary hospitals with imaging capabilities. And imaging in children, even in research, are rarely high resolution. So it's important to consider that it's a methodological constraint, but it can also be an advantage in terms of whatever connectivity or structural measures that you develop can be viewed through the lens of translatability as well. So we started off with this data set by looking at the basics. You can pre-process in a variety of softwares these days. In this case, the fundamental use of SPM or FreeSurfer to examine things like volumetrics and grey matter is a good starting point. And further on, looking at things like diffusion-weighted imaging and extracting measures such as fractional anisotropy and voxel-based morphometry are really useful. One of the key questions, I suppose, to ask at this stage is what aspects of the brain injury interact with the age of the child and what is unique to the injury itself. And we do know that things like grey matter volumes interact with age, so it's important to consider that the effect of age and how that's adjusted for in subsequent analyses. It may be that you want to include age as a continuous variable or a categorical variable. Most studies would employ age as a continuous variable and just regress it out. But you could dichotomize age into younger and older child groups, depending on your question of interest. Whether you adjust for age and sex and determine the effect of these variables post-hoc is entirely dependent on the research question that you pose. But if you are hypothesizing that there's a general structural or functional network based on these effects, then these demographic variables are important to include as confounders early on in your analysis. And that applies to other types of variables such as volumetric changes as well. So for this cohort, we focused on the use of voxel-based morphometry. We didn't find any significant changes in volumetric measures, but using the T1 scans and VBM, we first aimed to characterize grey matter changes in post-concussion syndrome in these MTBI children. And one of the key complaints that we focused on was the presence of poor sleep and fatigue, because we know that poor sleep and fatigue in these children lead on to poorer cognitive and attentional difficulties. So a VBM pipeline was developed, and for those familiar with segmentation software such as CAT, it's important to consider here age-based segmentation rules. In particular, software such as SPM generally defaults to adult tissue probability maps and normalization schemes. So if you're working with a pediatric cohort, I strongly encourage you to develop and use pediatric tissue probability maps based on the age profile of your kids. And not only does this provide better minimization of false positives, but also a greater specificity in delineating truer brain network effects. And what we found from the VBM results was that lower indices of grey matter were linked to poorer sleep. And all of this was controlled for age and sex. And in particular, this exercise was an important one as it showed us that a whole brain analysis, as opposed to a seed-based analysis, reveals some really strong convergence towards one particular region, being the default mode network. And this was one of the strengths of the study in employing this whole brain approach. And what we found was that these changes to the DMN negatively correlated with sleep symptoms. We then built upon this evidence from our T1 scans to explore functional changes within this network. And we applied a whole brain analysis to use regional homogeneity. And what we found was that this exact node in the posterior cingulate was also significantly negatively correlated with sleep. And at this point, we actually looked at the seed level functional connectivity between the DMN nodes. And what we found was basically that across these three measures, there was a very strong convergence of poor sleep and reduced connectivity of the default mode. Now, if we were to look at functional connectivity with a bird's eye view of whole brain fMRI, we were faced with certain challenges. Because we did have a smaller group of controls, and these controls were not necessarily controls that you would find in a hospital, but controls from the general population, we were faced with this disparity between a pathological group and a normal group, and a large pathological group, sorry. And what we found was whole brain network analysis didn't reveal anything significant. And we had several analytic theories as to why this may be. For example, group level comparisons can produce weaker effects, and with brain injuries such as MTBIs, which are already at the lower end of the injury spectrum, differences in brain networks may be more subtle than the symptom presentations themselves. We therefore tried a different approach. Rather than looking at whole brain networks, we decided to reduce the dimensionality of these brain networks by using canonical correlational analyses. And what we found was that this reductionist approach could link functional connectivity representations to a symptom and behavioral axis. And we found, for example, that lower default mode connectivity corresponded with poorer sleep and also poorer cognition, but also networks such as the visual and somatosensory networks were also related to these behavioral effects as well. And at this point in time, we'd arrived at a set of simplified metrics that told the story that the default mode network, just by using T1 and fMRI scans, gave us some sort of predictive value towards looking at recovery at the 3-month mark. From here, we wanted to go a little bit further by integrating diffusion-weighted imaging and tying in some of our findings from functional connectivity, but at a higher resolution to generate a closer one-to-one mapping of structure and function. And in particular, structure-function coupling in the sense of how strongly dependent the child MTBI brain structure informs function, and vice versa, was of interest here. We used the framework by Preti, published in Nature Communications. And one of the advantages of this framework was that its use of null models, which enabled us to disentangle the relationships between structure and function dependencies. And it also enabled us to distinguish whether coupling and decoupling effects are functionally or structurally reliant. A methodological consideration here is the use of the Glasser multimodal parcellation, which allows us to reslice to the fMRI resolution using the same parcellation that's used to construct the structural connectome from diffusion-weighted imaging. Now, I'm wary of time here, so I won't go into all of the results of the investigation, but some of the observations that we found with this work was that decoupling indices do indicate some pretty significant changes with cognitive scores. For example, if we look at cognitive tests of these children alongside structure function changes, we see that there's a decrease in function being coupled from the structural connectome itself, which gives rise to attentional problems. and also poorer inhibition, and also reduced reaction time. And this was observed in regions such as the prefrontal cortex, fusiform areas, and the temporal cortex. Now, notably, one of our observations is that these decoupling indices decreased, and that perhaps suggests that functional connectivity at these particular structural nodes were lower, and that indicates a dependence in white matter integrity at these nodes following an MTBI. And interestingly, these relationships in the prefrontal cortex seem to exist along some axis of structural functional decoupling with cognitive attentional problems. Okay, so to conclude, there were a fair amount of lessons that I particularly learned from exploring pediatric neuroimaging, and some of the ones that strike out at me are, for example, starting with the basics, so things like VBM, diffusion weighted imaging, like functional, fractional anisotropy, fMRI connectivity are good places to start. Using age-appropriate analyses and the types of confounders that you need to consider. Is your imaging protocol adequate for the type of analysis that you're applying? To perform whole brain or seed-based analyses. And again, this depends on the research question that you have and also the injury type that you may be dealing with, whether it's a diffuse injury or a focal injury, such as a stroke, for example. For focal injuries, you might opt for more seed-based analyses. And other factors such as control group availability and selection. For pediatric injuries, it's been suggested that the best group for comparing pediatric brain injuries is actually a group of children in the hospital who don't have brain injuries, such as orthopedic injuries. And other factors such as dimensionality reduction, and also when you're looking at structure function, for example, homogenizing between parcellation schemes as well. So these are all lessons that I've learned along the way, but there may be other factors to consider as well. But I'm very happy to take on any questions that you may have, and please contact me if you'd like to probe anything more from me. Thank you very much for your time.